Hello, everyone, and welcome to today's webinar titled A Tale of Two Techniques, Why AFA Shearing Outperforms Enzymatic Fragmentation in NGS Workflows. I am Sydney Panilla of LabRoots, and I will be your moderator for today's event. Today's web seminar is presented by LabRoots and brought to you by Covaris. To learn more, visit covaris.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for at the end of the presentation. You may also use the Ask a Question box to let us know if you're having any trouble seeing or hearing the presentation. Okay, I would now like to welcome our speaker for today's webinar, Dr. Lisette Payero, Field Application Scientist, Covaris. Dr. Payero, you may now begin your presentation.
Thank you so much for that introduction. My name is Lisette Payero, and I'm a field application scientist with Covaris. And today, I'm really excited to be able to bring you a tale of two techniques, why AFA shearing outperforms enzymatic fragmentation in NGS workflows. So we're going to be bringing you this story in 3 parts today. Part 1 is going to be an overview of next-generation sequencing, NGS library prep, fragmentation methods, requirements, and pitfalls. Part 2 is going to be a direct comparison of adaptive focused acoustics, AFA, versus enzymatic processing in NGS workflows, really comparing some real-world performance of the two methods. And then finally, for part 3, I want to take you beyond enzymes and highlight some unique applications where AFA is really uniquely poised to enable workflows. So let's start with NGS, next generation sequencing. I don't think it's an exaggeration to say that NGS has been a revolution in biological research. Unlike previous sequencing techniques, NGS enables massive parallel sequencing of millions of heterogeneous DNA fragments. And with these tools, researchers can get information-dense maps from their genomic samples, assembling genomes, detecting variants, and really making more informed decisions for both their clinical and their academic research. And today we'll be talking a lot about DNA fragmentation, a really essential step in NGS workflows. Now, this step is essential because sequencing platforms don't sequence whole intact genomic DNA, necessitating the need to shear the DNA down into smaller pieces or fragments. And it's these fragments that can then be barcoded, pooled, and loaded onto the sequencer for analysis. Now, fragmentation parameters and requirements can vary based on your workflows or your platforms, but there are a couple of guidelines that we can keep in mind. So when we talk about optimal fragmentation, we're really talking about 2 key metrics, and we're going to come back around to these pretty often. First, you want shearing that is uniform. That is, you want your fragments to fall within a narrow, ideal size distribution, with few fragments in your library that are substantially bigger or substantially smaller than your target. Next, you need shearing that is representative, meaning that the fragments generated meaningfully reflect the input sample. We could also call this random or unbiased shearing. So these are the 2 metrics that I'd like you to keep in mind today: uniform random fragmentation. And in order to achieve this, researchers have a couple of tools at their disposal. The first is enzymes. So enzymatic fragmentation, like you could imagine, leverages the catalytic mechanism of enzymes in order to cleave the DNA into smaller pieces. Now, since this is an enzymatic reaction, the enzyme needs to access, bind, and catalytically cleave the substrate, meaning that these reactions tend to fall within an ideal substrate concentration. And this is usually defined as an enzymatic unit. You might have seen this when looking at commercially manufactured enzymes. The enzymatic unit defines the right rate of substrate conversion. Now, in addition to being sensitive to concentration, enzymatic reactions can be sensitive to things like sequence bias, ideal reaction temperature, and also factors such as ideal enzyme storage. So the second method we're going to highlight is mechanical shearing, and in particular, acoustic sonication. So acoustic sonication leverages the physical properties of acoustic energy in order to shear your samples. Now, this is a really interesting process that may not immediately be intuitive. So when acoustic waves move through a liquid, they cause the microscopic gas particles within that liquid to form expanding cavitation bubbles. And these bubbles are going to expand and expand and expand until they reach a point of collapse. And it's this collapsing that actually produces the shearing forces that process your samples. So I'm going to show you a video here. And this video is just one cavitation bubble. But you can imagine that when you're sonicating your samples, it is the cumulative effect of hundreds and thousands of these bubbles that are processing your sample. So by manipulating acoustic energy, we can provide random tunable shearing that is independent of sample concentration. And at Covaris, we have really optimized this acoustic sonication with our adaptive focused acoustics, or AFA. Now, this is the technology that is at the heart of our ultrasonication instruments. Now, every Covaris ultrasonication instrument has a component known as the transducer. And the transducer is going to deliver acoustic energy in a geometrically defined pattern through your water bath to hit the focal zone of your Covaris consumable. And in this way, we can deliver precise, reproducible bursts of energy to your sample. Now, Covaris ultrasonication instruments are a little different from sonication instruments that you might have used in the past. One feature is that ultrasonication happens at the ultrasonic range, which is outside of the audible frequency for humans. For researchers, this means that you don't need to wear ear protection when you're using a Covaris ultrasonicator. The second feature is thermal control. So an unfortunate side effect of many traditional sonication methods is the introduction of heat to your samples. And you take a look at some of these thermal images of sonication, you can really see how this is the case. So when you have either a traditional water bath or probe sonicator, you can see that the samples are getting subjected to this kind of gradient of heat, reaching temperatures up to 40 degrees Celsius in some cases. Now, this is not ideal, especially when you're working with sensitive or volatile biomolecules. You can compare this to the Covaris sonicated samples, which are not getting subjected to this gradient of heat. So by precisely controlling the acoustic energy as well as the temperature of the water bath, Covaris is able to offer thermal control during sonication. So now that we know our players, we can begin to compare their performance. And so I'd like to talk to you about AFA versus enzymes in NGS workflows, taking a look at how well these methods deliver on the goals of uniformity and randomness that we've established. And let's start by looking at fragment uniformity. Now, NGS platforms are optimized for specific fragment sizes, and achieving a uniform size range is really crucial. It helps ensure that your library will cluster evenly and efficiently on the flow cell. Non-uniform shearing can be quite problematic because the presence of fragments that are too small within your library will cause an amplification bias. These shorter sequences will cluster more efficiently on the flow cell. And in fact, you can see this data here showing that amplification bias can lead to smaller insert sizes regardless of your fragmentation parameters. On the other hand, the presence of fragments that are too large will not cluster efficiently on the flow cell and end up being underrepresented in your data. So either way, a lack of shearing uniformity results in a loss of library complexity and library diversity. And so hopefully you can appreciate why shearing uniformity is a really important metric to look at when we're evaluating library preparation methods. And we actually have some pretty interesting data on this. So here we have data that's been discussed previously in another Lab Roots webinar with Quest Diagnostics. So if you're interested in taking a closer look, you can utilize that QR code in the corner. But let's take a look at some of this data. So in the first figure here, we have a look at the average insert sizes for libraries shared using enzymatic fragmentation as opposed to Covaris ultrasonication. And what we can appreciate is that while both these libraries actually do center around the target mean insert size, the enzymatically shared library is subject to this huge spread, with insert sizes both well above and well below the target. As we just established, this is going to be problematic because it means this enzymatically fragmented library is not going to cluster equally and evenly on the flow cell, leading to data skew or data loss. On the other hand, you can take a look at the Covaris shared library, and you can see that it has a much more uniform distribution of insert sizes, with the entire library falling neatly within the target parameters. This is going to lead to a better sequencing result. So that figure is a really nice example of uniformity within your libraries. I can also show you the second figure showing uniformity between libraries. So the second figure also shows you the mean insert sizes, but this time it's from libraries using a wide variety of input types for their DNA source. So what you can appreciate here is that by using Covaris ultrasonication, you can get uniform insert sizes, whether you're looking at buccal sponge, cell lines, cultured amniotic fluid, or saliva. You're able to get this really great consistent uniformity using many different sample types. And uniformity can have impacts upstream of sequencing as well. Whole exome sequencing, for example, relies on hybrid capture to enrich target genes. And we can see in this bottom figure here that the fold enrichment achieved with Covaris ultrasonication as opposed to enzymatic shearing is about threefold. And this really shows you that this process of enrichment is actually quite sensitive to fragment size. If you want to take a closer look at the underlying mechanism here, you can take a look at this second figure, which shows research indicating that enrichment in whole exome sequencing is really sensitive and dependent on insert size, with enrichment actually going down above and below the target size. Finally, we can also see how shearing uniformity contributes to sequencing metrics. So here we have data showing the discordance for libraries fragmented using enzymatic fragmentation as opposed to Covaris AFA shearing. And discordance can be a really interesting metric to look at because it's indicative of your overall library quality. And what we can appreciate here when looking at the enzymatically sheared libraries in blue is that they have a very high rate of discordance overall, reaching up to 15% discordance. It's also interesting to note that this discordance seems to be sensitive to the input amount. So we can see that as we go from 200 nanograms of input to 10 nanograms of input, the discordance is actually going up. So your library quality is suffering as your input goes down. In contrast, we can take a look at the Covaris shear data in orange. And we can see first off that the discordance is much, much lower overall, 2% as opposed to the 15% we were previously getting. It's also interesting to note that this discordance does not seem to be dependent or sensitive to the input amount. So you can see that we're getting very consistently high quality, whether the input is 200 nanograms or all the way down to 10 nanograms. So I think cumulatively, this data shows how a lack of shearing uniformity can impact both the efficiency and the quality of your library prep. The next metric I'd like to look at is fragmentation bias. So random shearing is really crucial in creating representative NGS libraries because NGS mapping relies on sufficient overlapping coverage of all of your regions of interest. Now, if you have fragmentation bias, this can create over or underrepresented regions in your data. And for researchers, this can mean additional or redundant sequencing as you're trying to fill in some of these gaps. So fragmentation bias is something that we'd like to be very mindful of. And we have some pretty interesting data here from Zach Herbert and Mara Berkeley showing the fragmentation bias of a variety of library prep workflows. Now, what we're looking at here is the per base sequence content. This is a metric that you'll often see generated from your sequencing analysis tools, such as FastQC. Now, all this is a plot of the proportion of each of the four nucleotides, A, T, G, and C, at each position across all reads in the input sequence file. And when you're looking at this data, what you want to see is a plot that looks like that top Covaris shared plot. You can see that you have these parallel even lines, indicating that the composition of nucleotides is not changing based on your position. And this is a good indicator of random shearing. Now, on the other hand, if you take a look at those bottom two plots that represent the enzymatically fragmented libraries, you see a much less consistent and much more jagged kind of pattern. And this is a clear indication of enzymatic fragmentation bias. So fragmentation bias can be a big problem, not only because it leads to a less beautiful per base sequence content graph, but also because it impacts gene coverage. So here we have some data showing how enzymatic fragmentation bias can contribute to over and underrepresentation in your sequencing data. So if I could direct your attention to this first figure, this shows the average normalized coverage of 25 cancer genes, genes that are commonly looked at in oncology panels. And what we have here is normalized coverage as a function of the percent GC content of the gene. So we can compare how different library prep suppliers are able to cover these 25 genes. And let's first look at this competitor in orange here. So you can see this in this orange line, it represents a library prep supplier that uses enzymatic fragmentation. And what we can see is a very clear skew. So we can see that normalized coverage is actually very, very high in low GC content genes. But as the GC content increases, you can see that the coverage is actually going down, indicating this really distinct preference for GC poor regions. Now, this is not ideal because it means that all of your genes are not getting equal amounts of coverage. What you would hope to see instead is something that looks a little bit more like that dark blue line that indicates the Covaris shared libraries. You can see that this line is pretty flat and parallel, indicating that your coverage is not really being impacted by the GC content of the gene. Now, if you're more mathematically inclined, this can also be described as an R-squared value or correlation coefficient in that little embedded table there. So you can see that the really high number that you're seeing for the supplier in orange indicates a very strong relationship between coverage and GC content. On the other hand, the low number that we're seeing for Covaris indicates that there is no such strong relationship between the normalized coverage for a gene and its GC content. If you're more visually inclined, you can also take a look at the second figure, which is a density plot, again showing the average normalized coverage of these 25 cancer genes. And what we can see here is that this supplier in orange, this enzymatically shared library, has this very broad, flat kind of peak. And what this indicates is that some of these genes are getting very overrepresented in your sequencing data, and some are getting very underrepresented, and the rest are kind of somewhere in the middle. Now, if you compare this to the Covaris peak in blue, you can see that it is a very tight, sharp peak centered around a normalized coverage of 1. And this means that all of your genes, your 25 genes, are getting equal amounts of coverage, again, regardless of their respective GC contents. Another interesting metric to look at when comparing library processing is variant detection. So here we have some interesting data showing the variant detection precision of a wide variety of library prep suppliers. And precision here is a measure of the false negative rate. So the higher the precision, the fewer false negatives are observed in your library. And this data is pretty interesting because you can actually compare and contrast the variant detection precision for all of these suppliers for both indels, insertion deletions, and SNPs, single nucleotide polymorphisms. So let's start by drawing your attention to supplier I in orange. So again, this is an enzymatically shared library. And you can see that the supplier is actually able to deliver pretty good precision when looking at indels. However, when we switch our attention to figure B, we can see that there's this drop-off in precision for SNPs. So there's this kind of difficulty in consistency when delivering precision between these two different kinds of variants. On the other hand, what we would hope to see is something that looks a little bit more like the Covaris data. So you can see that for both figures A and B, Covaris shared libraries are able to offer a high amount of precision, both for indels and SNPs. Now, interestingly, we can actually directly link how these differences in precision relate to sample processing, as supplier I also displays much less uniform shearing than Covaris libraries. And this is something that you can see in the DNA distribution in figure C. Now, you might take a look at this data and specifically take a look at the y-axis of figures A and B and wonder what the real impacts are of these minute differences in precision. And one thing I'd really like to emphasize is that when you're working at large scales, every decimal point counts. So if we were to look, for example, at supplier W in figure A, we can see that this 0.25% decrease in precision across something like 600,000 indel calls would be about 1,500 false calls. Similarly, in figure B, this 0.3% decrease in precision that we see with supplier I across 3.5 million SNPs is about 10,500 false calls. And so I hope you can appreciate that it's really our prerogative to optimize precision everywhere and any way that we can. And you can see the real impacts of using Covaris shearing by looking at a recent webinar from the Genome Diagnostics Lab at Radboud UMC. So as a core facility, they were facing the challenge of increasing requests and varying and diverse sample types. And they were able to meet these demands by incorporating Covaris shearing into an automated NGS workflow, enabling high throughput whole exome and whole genome sequencing that was flexible to sample type and composition as well as concentration, all while maintaining their sample turnaround time. So I think it's really important to highlight the places where optimizing these tools is really allowing labs to solve real-world problems. Okay, so up until now, I've really focused on comparing AFA and enzymes in NGS workflows. For this last part of my talk, I'd really like to hone in and highlight a few applications where AFA is really uniquely poised to enable workflows. And I'd like to start by talking about TrueSight Oncology 500, or TSO 500. So this is an NGS panel developed by Illumina to provide comprehensive profiling of cancer from FFP tumor samples. Now it targets over 500 genes to identify both DNA and RNA variants. This is a very powerful panel. It provides comprehensive data that is used to better inform patient care decisions. However, in order to be performed successfully, TSO 500 has two very important requirements. The first is high-quality nucleic acid, both DNA and RNA. And the second is precise, random, and uniform shearing during library prep. And I'd really like to highlight how AFA is able to deliver on both of these requirements, starting with nucleic acid extraction. So even though we've been talking about AFA in the context of DNA shearing, sonication is a very versatile tool, and it can be used in a wide range of workflows. In order to enable TSO 500, we can really highlight Covaris's TrueExtract FFP Smart workflow. Now, this workflow streamlines nucleic acid extraction from FFP samples, allowing parallel extraction of both DNA and RNA from the same FFP samples. Additionally, this workflow leverages AFA technology to enhance paraffin removal and tissue homogenization, doing away with the need for harsh organic solvents such as xylene. And this last feature is really important, especially for labs who would like to do these extractions at large scales using automation. Now, by incorporating AFA into these workflows, we can not only streamline these extractions, but actually produce better results. So one of the very important metrics for RNA extraction is known as the DV200. And this is a measure of the proportion of extracted RNA that is 200 base pairs or longer. For TSO 500, the DV200 requirements are 20% or more. And while that may not sound like a lot, it can actually be quite a difficult metric to reach using traditional workflows. So here I have some data from Discovery Life Sciences showing a comparison of their DV200 scores using traditional methods versus Covaris workflows. And this is a pretty striking figure. So I hope you can appreciate that using traditional methods, they were really struggling to get usable RNA, with only 6 out of 79 of their samples actually producing a DV200 score that met this threshold. In comparison, you can see that the Covaris workflows consistently produced quality RNA, with 90 out of 90 samples meeting and exceeding that DV200 threshold. Significant result. When doing TSO 500, you really want to be able to maximize the most out of your precious samples, ensuring that every sample possible is able to receive the full workflow. So that was some really powerful data that demonstrates how Covaris can enable TSO 500 through quality nucleic acid extraction. I also want to highlight that Covaris is relied upon to share TSO 500 libraries. So the TSO 500 protocol requires a very strict size range of 90 to 250 base pairs. And their guidelines actually highlight Covaris for this application, as enzymes are not capable of consistently delivering these results. So this figure here shows the fragment size distribution of Covaris shared samples. And you can see just how uniform this size range is, with all the samples falling within the required parameters. So this highly reproducible fragmentation profile is important, and it helps researchers consistently reach their targets. So between that data, hopefully you can see that combining high-quality nucleic acid extraction with precision AFA shearing enables TSO 500 workflows. And you can really see this reflected in TSO 500 sequencing metrics. So here I have data from our recent SLAS paper showing the sequencing metrics for the TSO 500 workflow for both DNA and RNA. And what you can see here is that by using Covaris methods, we can consistently meet and exceed TSO 500 metrics. This data was also collected using our automated assay-ready workstation, highlighting that Covaris can enable TSO 500 at high volumes while maintaining consistent quality. And you can really see the impact of this quality when looking at case studies from real labs. So here we have data from LabCorp showing the results of their in-site tumor profiling tests, both before and after implementing Covaris workflows. And what we can see is that prior to incorporating Covaris FFP workflows, full report rates were as low as 82% some months, meaning that up to 18% of patients were not able to receive full profiling from their tumor samples. After implementing Covaris, the full report rate increased to a consistent 95% and higher. Now, being able to receive a full report is a very significant thing. It means that no samples are getting left behind because of inoptimal processing. We can also demonstrate the potential savings that these results can bring to laboratories. And while all samples and all laboratories will experience different processing and different success rates, we can take a look at our results and compare them to previously published results from other suppliers to project and model potential savings here. And so hopefully you can see that by implementing Covaris workflows in TSO 500, you can have huge savings in both extraction, library prep, and sequencing. And so by using AFA, researchers are able to consistently meet and exceed the demands of their customers, all while working within the constraints of budget and time. And you can really see this reflected in our representation across many different somatic workflows. So here's just some of the market recognition we've received for enabling these assays. I think our presence here really highlights the way researchers rely on Covaris to process their most valuable samples. And speaking of valuable samples, we can look even beyond standard NGS workflows towards the arena of epigenomics. So here we have data comparing chip signal for samples sheared using a Covaris ultrasonicator as opposed to a traditional water bath sonicator. Now, anyone who's done chip before knows that it can be a very laborious process. It leverages formalin fixation to preserve interactions between DNA and proteins of interest. And unfortunately, if at some point during your workflow, the epitope of the protein is compromised prior to pull down, this can result in loss of signal for the researchers. Regrettably, this is a pretty common occurrence with traditional sonication techniques, as traditional sonicators can introduce an undesirable amount of heat to the samples and be quite harsh. And we can really see this reflected in the data here. So by looking at the water bath sonicator in orange, we can see that these samples yielded a very low chip qPCR signal, indicating that the epitope was compromised during sample processing. In contrast, we can see that the Covaris sonicated samples were able to yield a much higher qPCR signal, indicating that using Covaris ultrasonication, we were able to consistently shear the chromatin while maintaining epitope integrity. So I wanted to highlight this data because it shows that chip, like many workflows, really benefits massively from the controlled, precise, and thermally regulated processing that AFA can offer. Another workflow that really benefits from Covaris AFA is Hi-C. So Hi-C, much like chip, utilizes fixation to preserve interactions, in this case, chromatin-chromatin interactions. And Hi-C libraries end up being quite interesting. So Hi-C libraries end up being composed of all of these unique fragments containing junction points that reflect places where chromatin interacts spatially in a cellular context. And in order to get the most out of this data and make high quality contact maps, unbiased reproducible shearing with tight DNA fragment sizes is really crucial because you want to capture all of these chromosomal interactions in order to get a good spatial picture of the chromatin. So AFA is used really widely in Hi-C protocols, with several papers even highlighting how consistently AFA is able to shear their chromatin during these workflows. Next, we can take a look at methylation sequencing. So methylation provides another important layer of epigenetic information. And methylation is being increasingly recognized as a valuable biomarker. It regulates chromatin structure, transcription factor binding, and ultimately gene expression. And methylation sequencing techniques tend to use either chemical or enzymatic conversion to identify methylated residues. So here in this image, we have a workflow that utilizes chemical conversion to convert all unmethylated cytosines into uracils, ultimately providing researchers with a kind of map of methylation sites. Now, AFA is capable of enabling methylation sequencing for a couple of reasons. So first, AFA shearing is agnostic to conversion method, working whether you are using chemical or enzymatic conversion. Secondly, and probably most importantly, AFA is able to maintain methylation markers that are sensitive to enzymatic fragmentation, really preserving your sample and preserving your signal. So for these reasons, Covaris AFA is very highly recommended by manufacturers of methylation sequencing reagents. Finally, I'd like to highlight cases where AFA can enable analysis of exceptionally difficult sample types. So one such sample type is circulating tumor or ctDNA. These are fragments of DNA that are shed by tumors into the bloodstream, and it's a really valuable biomarker in early cancer detection and disease monitoring. Unfortunately, this sample type is also very difficult to work with, making up an extremely small proportion of CF or cell-free DNA and often being of very poor quality. So this is a pretty striking figure showing just how small a proportion of the total cfDNA can be composed of this tumor-derived ctDNA. And the authors even point out that an average patient sample can have as few as 15 total molecules of the ctDNA. So when we're working with the cutting edge of detectability, getting the most out of these samples is really crucial. And AFA is relied upon in these workflows to provide high quality, comprehensive whole genome sequencing maps from blood samples. These provide the kind of baseline mutation information that can then be used when comparing your ctDNA sequences. Next, AFA is also relied upon to provide precise shearing of small spike in controls that are essential in the sensitivity of these assays. So it's really gratifying to see the way that tools like AFA are facilitating workflows that are really pushing the limits of sequencing sensitivity. And with that, I'm going to leave you with a couple of conclusions. So NGS library workflows are demanding increasingly flexible, precise, and consistent sample processing. And hopefully today I've been able to show you that Covaris AFA delivers uniform random shearing that outperforms enzymes in library preparation. I've also shown you that AFA maximizes sample recovery, data quality, and workflow ease in traditional NGS applications, as well as enabling researchers to expand their workflows, including low abundance, unstable, and complex sample types. Now, before I leave you, I did want to quickly plug our latest offering, which is the Covaris TrueCover Whole Genome Sequencing PCR-free Library Prep Kit. In my talk today, you've seen how Covaris has worked upstream of these next generation sequencing protocols for many years, providing the gold standard of DNA shearing for NGS applications. So we're really excited to be able to offer this kit and support our customers post-shearing, taking ownership of the entire library prep process. Great. And with that, I would like to thank you all for joining me today. I would also like to thank and highlight some other Covaris team members for their support and their expertise. And here we have some of our contact information as well for those who might want to reach out and have questions after this talk. And with that, I'll take any questions. Thank you very much.
All right. Thank you very much, Dr. Pajero, for your very informative presentation. We will now go ahead and move into the live Q&A portion of the webinar. If you happen to have a question that you would like to ask, please do so now. Just type your question into the Ask a Question box and click Submit. We will answer as many of your questions as we have time for. Okay, let's get started here. The first question that we have says, what buffer can be used for AFA?
Oh, yeah. So that's a great question and a very fundamental question. We do recommend for DNA shearing to use 1xTE, which is a buffer that is very accessible. I'm sure most people working in labs have used it before. And we do recommend not to shear in plain water, as you do need the buffering capabilities of something like TE when you're shearing DNA.
Perfect. Thank you. The next one that we have here says, what is the cost of AFA?
Sure, that's also a great question. Thank you. So we do have a range of ultrasonication instruments depending on the needs of your lab. When we talk about the cost of AFA, I think it's really important to take a holistic approach that includes the cost benefits of your sample processing being improved. So by increasing your efficiency in sample processing, what we're really able to see is that people are able to decrease their failure rates and decrease the rate at which they have to rerun their sequencing. And when you take that into account, you can actually get some very substantial cost benefits from these kinds of workflows.
Perfect. Okay, our next question here says, you discussed Illumina's TSO 500 as an important application of AFA technology. I noticed you had a slide with several other oncology workflows that also use Covaris. Have you seen similar results with those, or are there any that stand out to you?
Oh yeah, absolutely. That's a great question. We show a whole bunch of those different workflows, and I think I had a slide that actually highlighted some of the many oncology workflows that we are a part of because we are widely relied upon in that space to provide like comprehensive and reliable sharing. So I do like to highlight all these different workflows because one thing that they share is the need for robust and reliable sharing, and that's what Covaris AFA can offer. So you can see that reflected in the TSO 500 data that I presented to you today, but you can also see that highlighted in other workflows such as PGDX profiling.
All right, fantastic. Okay, it looks like we have one final question here. And this one says, can AFA be used for proteomics?
Oh, yes. So that's also a fantastic question. So the scope of this talk today really focused on DNA sharing, and I was also able to touch just very briefly on nucleic acid extraction. But we do also offer proteomics solutions and proteomics workflows. And that's going to be whether you're using FFP, fresh frozen tissue, or cells, right? You can use Covaris AFA to enable your proteomics workflows as well. And there are some unique benefits to using AFA for proteomics. One, that comes to mind is again our like reliable thermal controlled processing is really great if you are trying to maximize your protein integrity or maintain your native protein activity.
All right, wonderful. Thank you. Well, thank you again, Dr. Pajero, for your time today and for your very informative presentation. We would also like to thank LabRoots and our sponsor, Covaris, for underwriting today's educational webcast. Before we go, I would like to thank our audience for joining us today and for their very interesting questions. Questions that we did not have time for today and those submitted during the on demand period will be addressed by the speaker via the contact information you provided at the time of registration. This webinar can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, everybody, goodbye.